Okay, so you guys can read the objectives on your own time because we're going to be pressed for time tonight. And so, what is muscle fatigue? You can have two basic definitions, right? They are what? What are the two definitions of muscle fatigue? Okay, a decrease in muscle force production. Okay, when I say muscle, I'm referring to the whole muscle. Okay, and if you have a decrease in whole muscle force production, then you're going to have that due to a decrease in myofiber force production. Okay, what was our other definition? Well, we'll phrase it as increased effort, okay, which will be increased motor unit recruitment, increased effort to maintain the same work rate or power output. Okay, and first one you probably, that's pretty common definition. Second one, may or may not have seen that one before, but it's true, right? I mean, if you're having to work harder, if you're having to dig deeper to maintain your velocity, then your effort has been increased to maintain a given power output, and if that's the case, then you are in a state of fatigue. Okay, and that's because some of the muscle fibers, their ability to produce force is reduced, and you're having to recruit more motor units to compensate. Okay, so in that case, some of the fibers some of them have force production impaired. All right, so everybody know who this is? Usain Bolt. And so does this man fatigue in 100 meters? Okay, so he's sprinting 100 meters. Does he fatigue? What's that? No. Who says no? Okay, who says yes? Okay, and this is when he ran 9.58, which I think is the world record. You can see that his highest velocity was achieved between 60 and 80 meters when he was running roughly 28 miles per hour. In his last 20 meters, he was only running basically 27 miles per hour. So he was slowing down the last 20 meters of that race. Evan? Well, you typically, if you're a good sprinter, you celebrate afterwards. But when he ran 9.58, his world record, he did not celebrate before the finish line. He did, I'm sure he did after crossing the finish line. That's when the clock stops. Oh yeah, he definitely will showboat on some. But when you've got $500,000, $1 million bonuses right in whether or not you set a world record, you're going to try to set a world record. And so, you know, he's definitely trying through the whole race. And this basically applies to everybody, okay? And so most people are going to be slowing down the last portion of a 100-meter race, a 200-meter race. It's who's slowing down the least and who's been able to maintain the highest velocity up till that point, okay? So even in sprinting, an event that lasts 9 seconds, 10 seconds, maximal power output, you're going to experience fatigue, okay? And so do these guys fatigue in the metric mile 1,500-meter race? Does it look like they're fatiguing? Most definitely. Okay, and everybody's favorite race, the 200,000-meter race, okay, which translates to about 130 miles. And this particular race starts in Death Valley with 120-degree temperature and then finishes on Mount Whitney in California. Zoe? Well, it could be both, right? I mean, it depends. If they feel a twinge in their hamstring or something, they're going to slow down. But if they're not being injured, they're trying to win the race, and they're going to try to run as fast as they can through the entire race. You guys don't believe me? People fatigue in 100 meters? Believe me, everybody go try it tonight or tomorrow. You'll fatigue the last, hopefully, last 5 meters of the race if you're really fit. Okay, if you're giving it your all. But I mean, that's why I put that table up. Those are the actual variables in terms of time per 20-meter segment, and that way you can calculate it. And he's better than most people. He doesn't fatigue as much, and that's why he's so fast. Okay, but back to these guys. Okay, so do these guys fatigue in a 130-mile race? Most definitely. Okay, and so this slide illustrates the idea that, well, the last 3 slides illustrate that you can fatigue in sprinting conditions of short duration, moderate duration, and in endurance races of long duration, right? So muscles can fatigue, but is the cause of the fatigue exactly the same in each case? No. The likely cause of what causes fatigue in a 100-meter race is going to be different than what causes fatigue in this 130-mile race. And this slide also illustrates the fact that environmental conditions can certainly compound and contribute to the cause of fatigue. And so the reason I point that out is for the rest of the class tonight related to fatigue, we're not going to consider environmental factors. Okay, I'm sure you talked about that in exercise phys, where you talked about heat stress, okay, how that can affect cardiopulmonary performance, skeletal muscle performance. What we're going to take is just going to say generic, moderate duration, high intensity exercise and talk about fatigue in that case. And so if you want to say a 5-minute race, a running race for 5 minutes, then that's what we're going to be addressing. Okay, so 3 minutes, 5 minutes, 10 minutes, trying to go as fast as you can in that period of time, then that's the type of fatigue we're going to be addressing. Okay, so high intensity, but I would say moderate duration, not in the order of seconds, but not in the order of a day either. All right, so let's look at the characteristics of the muscle contraction. So when muscles are fatigued, again, what's the primary characteristic of fatigue of the muscle or of the affected myofiber? Okay, it's a decrease in force production. So maximal force output is going to decline. So with this figure, what you're looking at is an actual maximal muscle contraction of a fresh muscle and a fatigued muscle. And so this represents the peak force, okay? This probably corresponds to some isometric contraction. And so that represents the peak force, and then you can see in the fatigue state they're producing 20%, 30% less maximal force, okay? And by definition that's fatigued. Okay, what else do you notice about the fatigued muscle's ability to produce force? 2 other things. Very clear. Okay, the rate of force production is depressed in the fatigue state, so you can't apply the force as fast in the fatigue state, and the rate of relaxation is drastically affected. Okay, and so if you're thinking about running, cycling, swimming as hard as you can for 5 minutes, 10 minutes, that's contraction and relaxation. And so both contraction and rate of force development and relaxation are going to be affected. Okay, so peak force, rate of force production, and rate of relaxation. And so what you're going to see also is the Vmax, okay, the maximal shortening velocity of the muscle is going to decline. Okay, these will all decline. Okay, And so what else? If you're having peak force decline and the velocity of movement decline, by definition, power is force times the velocity of movement, and so you can see power is going to be reduced. And it's not just reduced. I want you to take away the fact that it's reduced a lot more than just the force or the velocity by themselves. Bless you. Okay, so that's shown here. If you're looking at this is a force velocity curve in a fresh and a fatigued muscle, and so this represents the fresh muscle here. Okay, so you're just basically looking at different shortening velocities and measuring how fast the muscle can, how fast it can shorten and how much force it can produce. And then what's on the y-axis is this is Vmax in the fresh state, and this is Vmax in the fatigue state. Okay, peak force, peak isometric force in the fresh state, peak isometric force in the fatigue state. Everybody see that? And so from a magnitude perspective, they're about the same magnitude of a drop in force and the same magnitude of the drop in velocity. And if you looked at that magnitude would put it about here if power was going to be affected to the same degree. But you can see power is reduced twice as much as either force or velocity, maximal velocity is concerned. And that shouldn't be surprised because power is a function of both those things. Okay, so power is a product of force and velocity. So if both those things are affected, then it's going to compound the effect on power. And power is the primary factor that's going to move you, okay, in terms of running, swimming, cycling, throwing, whatever event you're talking about. Okay, so besides peak force, rate of force development, relaxation, this slide shows you that maximal shortening velocity is reduced, power is reduced from here to here, and the third thing you have to know about this slide is that power is affected a lot more than either force or velocity alone. Everybody got that? The effect of fatigue on power is compounded by decreases in force and velocity. Okay, so if you're looking at the effects of fatigue on the fiber types, so what would you imagine? Type 1 fibers, slow twitch fibers, would they be affected more or less compared to type 2 or fast twitch fibers? Less, okay, and so the soleus, okay, is primarily slow twitch, so that's primarily type 1 fibers. Plantaris is primarily fast twitch, okay, type 2, and you can see in terms of these force velocity, very little drop in velocity, very little drop in force, and you can see very little drop in power. Okay, but certainly the drop in peak power is more than the drop in the force or the velocity by themselves. Whereas the fast twitch plantaris, you can see a pretty big drop in Vmax. Okay, this is actually normalized to force, so you can't really see it, but the force would be reduced. And you can see, again, a much bigger drop in power in the fast twitch muscle compared to the slow twitch muscle. Okay, and that's not surprising because we describe type 1 fibers as being fatigue resistant, right? Not being fatigable. And so this just illustrates if you stress, if you force slow twitch muscles to do, say, 1,000 contractions and force fast twitch muscles to do 1,000 contractions, the fast twitch muscle is going to experience a lot more fatigue, greater decline in force, greater decline in Vmax, greater decline in power compared to that slow twitch muscle. Okay, so when we talk about the inability of the muscle to produce force, then where can we see failure occur in the what I'd call the motor command pathway? Okay, so if you think in terms of the activity, then it's going to start with the brain. Okay, so we're calling this willed movement. So you're going out to race a 5K, race a 10K. You have to do that. You have to want to do it. So it starts in the brain. So again, primarily motor cortex and other parts of your brain are going to initiate that movement. Okay, and so from there, this sounds an awful lot like the excitation contraction coupling pathway, right? So from the spinal nerves, okay, you have efferent and afferent spinal nerves going from the brain down the cord, and they will synapse with alpha motor neurons that are going to control a given set of muscle fibers to a given muscle group. Then you're going to have a connection between the nerve and the muscle fibers. Then you've got to conduct the action potentials along the plasma lemma down the T-tubule, activate the dihydropyridine receptor, in turn activating the ryanodine receptor, causing calcium release from the SR into the cytosol. That calcium is going to bind to troponin, move tropomyosin, allowing for actin and myosin to interact and produce force. Okay, so this is the excitation contraction coupling pathway we talked about. So basically any spot, any site in that pathway is fair game in terms of where failure may occur, but in healthy adults the failure sites are going to be more limited. Okay, and those failure sites can be associated with the brain. Okay, not so much the alpha motor nerve, okay, not so much the neuromuscular junction, but you can have problems with, besides the brain, the T-tubules, the SR's ability to release calcium, and the cross bridges. Okay, so those are your primary sites of failure associated with fatigue. Okay, so we have the normal process, the normal pathway for activating the muscle. There are certain sites along that pathway that are more susceptible to failing, and these are the most common failure sites: the brain, the T-tubule's ability to conduct action potentials, the ability of the SR to release calcium, and the ability of actin and myosin to generate force. Okay, are you with me? So these are potential and likely sites of failure within the system, within the neuromuscular system. You should then ask, well, what causes these sites to fail? Okay, these are failure sites, but what causes them to fail? And so you're basically looking at your ability to deliver oxygen and nutrients to the muscle fiber as being one potential cause of the problem, in terms of having adequate fuel substrate. And that's where the glycogen is going to come in. On the other side of the metabolic equation is you produce byproducts in energy metabolism. And those byproducts can impair these sites. And so likely metabolites or byproducts of metabolism that can cause fatigue are going to be elevations in ADP, inorganic phosphate, hydrogen ions, potassium, and reactive oxygen species. Okay, does everybody see this? So you have these sites that can fail. They fail either because they're not getting, the cell's not getting enough oxygen and or nutrients, or it doesn't have enough fuel substrate in terms of glycogen, and or the cell's been working so fast in terms of the rate of energy consumption is so high that the rate of metabolite production is also high, and the system cannot clear or get rid of those metabolites. And those metabolites can interfere with these processes. Okay, so for the rest of the notes, we're going to be looking at these sites of failure and the cause of the failure, okay, in terms of either energy substrate depletion or lack of ability to deliver stuff to the cells versus energy metabolites, okay, byproducts of energy metabolism accumulating. Questions about where we are so far, this slide in particular? Okay, so in addition to that, we have afferent nerves that will signal not just, okay, we know that group, the 1A and 2 afferents are associated with what muscle receptor? Got to know it for the final. 1A and 2 afferents are associated with what muscle receptor? Muscle sensory receptor. Muscle spindles. 1B associated with Golgi tendon organ. Type 3 afferents are considered mechanical sensors. Type 4 are considered metabolic sensors. Okay, and so these afferent nerves feed back directly into the spinal cord and can synapse and affect the recruitment and the threshold of motor units, those alpha motor nerves, going to the muscle, but they will also send information to the brain. And this is very important because these affect blood flow, cardiovascular, cardiopulmonary centers of your brain. And if they affect the brain stem in particular, and the brain stem is where the cardiovascular, cardiopulmonary control centers are in terms of heart rate, contractility, okay, blood vessel diameter. Did you guys learn any of this in cardiopulmonary? Okay, so these afferents will project to the brain, and in particular to the cardiopulmonary, cardiovascular centers, and they will then influence blood flow delivery to the working muscles where these afferents originated. And if it alters the blood flow, then you're altering this side of the equation in terms of the muscle's ability to maintain contractile output, whether it's going to get more blood or less blood. Okay? So these afferents can directly affect threshold of alpha motor nerves in their ability to be recruited, but they will also affect the brain and cardiopulmonary, cardiovascular centers of the brain to affect blood flow, because you only have so much blood flow, right? You only have so much blood in your body, and your cardiac output's going to be limited, and so you can't increase blood flow to every organ in your body, right? It'd be nice to be able to say, let's increase it to our brain, increase it to our exercising limb muscles, increase it to our respiratory muscles, increase it to our kidney or our intestines, but that's not what happens. You have to redistribute the cardiac output to the working muscles that are critical for the survival of the organism. And so if you're exercising, you get less blood being delivered to the gut because digestion isn't necessarily important in that 5 minutes or 10 minutes that you're racing or potentially trying to escape predation. And so blood flow will be reduced to your gut. And it's going to be, and that allows more of it to be shipped and delivered to the working skeletal muscles. And that enhances your ability to run fast or your ability to survive. Okay, so these are very important in terms of how they affect your exercise and your fatigability. And we'll talk about them in a second. Okay, so we can break fatigue into two components, central fatigue and peripheral fatigue. And central fatigue refers to anything dealing with the central nervous system, so that's the brain, spinal cord. But we said there's not much happening in the spinal cord, okay, in terms of the motor nerves projecting down. They don't fail, okay, but whether or not they're fully activated will depend on basically how motivated you are. Okay, so I'm not going to go over it. This is another contraction, but central fatigue refers to the reason force production, power production is declining because it's a problem with the central nervous system, and in particular the brain. And typically these things are related to motivation, okay, emotional states. So if you start off kind of depressed, your favorite show was just canceled, and it's like, how am I going to survive? How am I going to continue? I got to go work out, but, you know, or I've got to go race, and you're just not feeling it, right? And so your ability to recruit motor units and to achieve a relatively high rate of rate coding depends on what's flowing out of your brain. And so if you're not feeling it, then that's going to affect your cortical outflow, and you're not going to recruit as many motor units or at a high rate coding level. Okay. If you're racing and you're like, this hurts, I don't want to do this, and so your motivation changes, right? And so pain makes fatigue a lot easier, and so your motivation, your pain threshold may modify, like, I'm not going to keep doing this, this hurts too much, and so you slow down, right? And so that's an example where your brain is dictating how many motor units you're recruiting, your power output, and it's going to decrease it. You're going to slow down. Okay, so those are examples of where the brain is limiting your performance. It's causing muscles to produce less force and power. Okay? Questions about how the brain can modify motor unit recruitment and rate coding based on level of motivation or your emotional state? Okay, now this addresses these afferents. Okay, and so this is a great slide. Wouldn't it be great if I had some labeling on the final exam and you guys get to label this? Let's go back a little memory lane again. 1A, afferents coming from the spindles. Okay, 2 coming from the spindles. And again, open circles, these are either interneurons. Okay, and if it's black, then it's inhibitory. If it's white or open, it's excitatory. And so you can see the spindles are going to activate the muscle. Okay, stretch reflex, muscle stretched, then those afferents will cause activation of the muscle. So the motor units are going to be excited. The alpha motor nerves are going to be excited. So these are excitatory. Okay, the 1B coming from the Golgi tendon organ, you can see it's going to synapse with an interneuron that's inhibitory. So Golgi tendon organs are inhibitory in terms of motor unit recruitment. And then you have these 3s and 4s. And so these 3s and 4s, again, can feed back directly onto the alpha motor nerve. And you can see they are going to inhibit the alpha motor nerve from being recruited, so you're not going to recruit as many motor neurons. But what's not shown is that these also project to the brain. These afferents project to the brain, and then the brain can project down to the alpha motor nerves. But importantly, they will project to vessels, blood vessels, and everybody remember smooth muscle associated with arteries, arterioles. Arterioles are going to be associated with being essentially in the muscle, and then those arterioles will branch into capillaries, and those capillaries branch into venules. Okay, and so the diameter of those vessels determines how much blood is getting to the muscle fiber, right? So whatever's downstream of that arteriole, okay, will get blood. And so if you have vasoconstriction of those arterioles, the vessel kind of clamps down and you reduce blood flow to anything downstream, including the muscle fiber. If those arterioles vasodilate, then you increase blood flow to the muscle fibers downstream. And effectively what happens is when these receptors become activated, they send that information to the brain, and the brain says, okay, there's a lot of metabolites being produced by this particular skeletal muscle, okay, in your thigh, in your leg, and it's too many metabolites. Blood flow, we can't increase blood flow anymore, and so we must reduce blood flow in order to save it for other areas, namely your respiratory muscles. Your ability to breathe is very important, not just to your working muscles, but to your brain and all your other organs. Okay, so there are studies that show that these afferent receptors will send information to the brain, and then the brain will then affect vasodilation, the levels of vasodilation or vasoconstriction to the working limb skeletal muscles, and it reduces it. Okay, and so it basically would reduce delivery of oxygen and nutrients and energy substrates to those working muscles, effectively facilitating fatigue of those fibers that are affected. But it's doing so in the sake of saving blood flow to respiratory muscles and presumably your heart. Okay, and so it's sacrificing your limb skeletal muscles and maintaining contractile ability of muscle fibers associated with your diaphragm and other respiratory muscles and your heart. And so if you're reducing blood flow to the exercising limb muscles, you're going to speed up fatigue. And so that's how these afferents can affect fatigue. Okay, again, it's because that information about stretch or metabolites sent to the brain, and then the brain will decide about blood flow delivery to those muscles. And if it reduces blood flow, it's going to facilitate fatigue to those muscles. Okay, so in a way that's going to be related to central fatigue. And by the way, if you're into racing and pacing, then these receptors play a primary role in deciding what pace you choose. What's the fastest, highest power output that you can maintain without compromising other organs, okay, essentially, or other systems in terms of respiration or cardiac function. Okay, so these play a vital role in pacing. Okay, central fatigue becomes more important as the duration of the activity increases. Okay, typically you don't have a lot of central fatigue in activity lasting 5 minutes, 10 minutes, 30 minutes. If you're talking about hours, then it becomes a greater problem in terms of your motivation, in terms of driving the motor units to work. Most fatigue is going to be what's called peripheral muscle fatigue, and so the problem is going to lie within the muscle. Okay, and so we said a likely site within the muscle fiber is the T-tubule. And the problem here, in terms of this is a normal action potential in terms of its amplitude and rate of depolarization, rate of repolarization. In a fatigue state, you can see the amplitude is reduced and the rate of depolarization is reduced, rate of repolarization is reduced, and so that could potentially impair your ability to activate the dihydropyridine receptor. Okay, and that change in excitability of the T-tubule membrane is caused by an increase in potassium inside the T-tubule membrane. Okay, if you remember, every time you activate the muscle, okay, if that muscle fiber belongs to a fast motor unit and it's being recruited 50 times a second, then that's 50 times it's being turned on and off within 1 second. Okay, so each activation requires an action potential, and that's going to require sodium moving into the cell and potassium coming out of the cell. This is a pump that reverses that distribution. Okay, and so imagine high intensity activity lasting 10 minutes. That's a lot of action potentials in that muscle fiber, and what happens is this potassium tends to accumulate in the T tubule. And when it does that, it impairs the action potential propagation, and that can impair activation of the DHPR, which everything downstream would then be affected. Okay, so this does happen. Potassium ions do accumulate in the T tubules with high intensity activity, and it can contribute to the decline in force, but it's not the major cause of the decline in force. Questions about T tubules? Potassium? Actually, that's an excellent question. What is the recovery for fatigue? Because we're going to talk about injury next, and that's one thing you guys must know, is the difference between the duration of fatigue and the duration of injury. So fatigue, what does your gut tell you? How long are you going to have this impaired force-producing capability? Say 5 minutes, you go out, 5 minutes, 10 minutes, 20 minutes, and you're fatigued at the end of this event. How long does it take for the muscles to regain maximal force-producing capability? Yeah, but that's energy delivery, and usually energy delivery in terms of ATP or creatine phosphate, that's not the problem, right? Because ATP levels return within a couple minutes, right? So how long is maximal force producing capability reduced with fatigue? Sure. It's more of a relative answer. Okay, it's on the order of what? Nanoseconds? No. Microseconds? No. Seconds? Maybe not seconds. Minutes? Yeah, you're talking minutes to hours, right? So within if you're talking about a 5K or something, then within 3 hours you're probably recovered in terms of maximal force producing capacity. Okay, so force is reduced on the order of minutes to hours with fatigue. You're talking minutes. Yeah. So it depends, right? So if you're talking about the guys that run for a day, 130 miles, there's fatigue, but there's also going to be injury there too. But if you're going to isolate the effects of the fatigue, it's still hours. Okay, it might be more than a couple hours in that case, but then it's getting convoluted with injury too. Okay, so this recovers within minutes or hours. Okay, this is just showing you how this is EMG and the amplitude of that. This just corresponds to a bunch of interference patterns while the muscle is activated, and you can see that it declines, okay, throughout 3,000 contractions. Okay, so there's no doubt that the ability of the membrane to conduct action potentials is affected with high volume work, high intensity work, okay? And so this does contribute, but again, it's not the primary cause of the reduction in force. So you're asking, what is the primary reduction? Okay, what is the primary cause of fatigue? And it is, I switched the order of a couple slides here, sorry. So it's just following the EC coupling pathway. So we go from excitation, action potentials to activating the DHPR, activating the ranitidine, releasing calcium. And so it's an impairment in calcium release is the primary cause of fatigue. Okay, in most cases, certainly most peripheral muscle fatigue cases, the SR within the muscle fiber is not releasing enough calcium. And if it's not releasing enough calcium, it's not binding to as much troponin, and so you're not moving as much tropomyosin, and so you're reducing actin and myosin interaction, which will reduce force production. Okay, so this just shows you a series of maximal contractions over the course of a few minutes, and you can see once the muscle drops force initially, and what's shown down here is calcium concentration. And so this is basically a contraction of the muscle, and this shows you how much calcium is released, and that's normal. And then during this period where force is reduced, you can actually see that force production or calcium levels are elevated. So calcium's not causing fatigue here, but once you see force production start to really be reduced, so again, each spike corresponds to the maximal contraction. And so once you get in here, okay, force is dropping. Okay, so the amplitude of this spike corresponds to peak force. So everybody see that that's fatigue, lots of fatigue here, where it looks like force is reduced maybe 2/3, 60 to 70% decline in maximal force production. And if you look at calcium, you can see calcium is reduced maybe by half. Okay, so in this case, the reduction in calcium release in the amount of calcium in the cytosol is what's causing this drop in force production. Okay, just to test that hypothesis, then what they can do is expose the muscle to a drug that bypasses all the other steps in the excitation process and directly acts on the ryanodine receptor. You may have not have known that, but caffeine binds to the ryanodine receptor and it opens it up. And when it opens it up, what happens to the amount of calcium? It goes up. And what happens to force producing capability? It goes up. Okay, so this shows that in this particular experiment that the reason force declined here from here to here was because the amount of calcium being released was reduced. If you can restore the calcium release, you effectively restore force producing capability. Okay, so this is the primary cause of weakness associated with fatigue. Again, this lasts for minutes to hours. Impaired cross-bridge mechanics in terms of actin and myosin interaction, that's also impaired in fatigue. Okay, so you don't need to know all these steps. This is just a bunch of steps associated with actin and myosin, calcium coming in, releasing phosphate, releasing ADP. Okay, take-home message here is that fatigue is also associated with a decline in cross-bridge mechanics. Okay, and the primary cause of that is an increase in inorganic phosphate. Okay, and let me go back to the calcium. The primary cause of the impaired calcium release is also due to an increase in inorganic phosphate. Okay, so an increase in inorganic phosphate, which happens every time ATP is hydrolyzed, okay, and there's a lot of things breaking down ATP, and the inability to get rid of that inorganic phosphate is going to cause reduced calcium release, and it will also cause impaired cross-bridge activity, cross-bridge mechanics. Okay, so you must be able to identify the site that fails and also tell me why it fails. And so for T-tubules' ability to conduct action potentials, that impairment is caused by what? The accumulation of potassium. Okay, every time the fiber is activated, there's depolarization and repolarization. Repolarization is associated with potassium efflux, potassium leaving the cell, and what's inside the T-tubules outside the myofiber. And so you're disrupting the ability of the excitation signal to propagate down into the fiber. Okay, the primary cause of fatigue is impaired SR calcium release. The thing that causes impaired SR calcium release is an increase in inorganic phosphates and a decline in glycogen. Okay, and it's not related to the ability to deliver energy for the actin and myosin process. It's some other reason that we don't know. Okay, studies have confirmed that if you reduce glycogen levels associated with the myofibrils, then it impairs SR calcium release. Okay, everybody follow that? Because that's an important distinction. Typically, we associate glycogen with the ability to supply ATP with the cell. You break down the glycogen through glycolysis and make ATP very quickly. Okay, the reason the SR can't release calcium, okay, related to glycogen is not due to its ability to supply the cell with ATP. It's some other thing that we don't know. Okay, so this is the primary site of failure, and it fails because of an accumulation of inorganic phosphate, but it also fails because of a decrease in glycogen within the myofiber, specifically within the myofibrils. Crossbridge fail primarily due to an increase in inorganic phosphate. Well, when is force produced? At the crossbridge, when is force produced? That's the power stroke. So what causes the power stroke? The release of inorganic phosphate from the myosin head. Remember, the myosin head will break down ATP into ADP and the inorganic phosphate. It's the release of inorganic phosphate that causes the power stroke, and that's where force is produced. And so presumably the increase in inorganic phosphate will impair the release of phosphate from the myosin head, and so you're not getting as much or as fast. The rate of release isn't as great, and so force per crossbridge will be reduced. Okay, so this is kind of a summary slide related to peripheral muscle fatigue, in the sense that if you follow the numbers, what's being shown here is force on the y-axis and calcium concentration on the x-axis, and so it's kind of biased towards SR, but it's also will address another factor causing fatigue or weakness, and that's sensitivity. Okay, so force, absolute force is reduced, and that force is reduced because there is an impairment in crossbridge mechanics, right? And potentially potassium accumulating within the T-tubule, okay? But the primary cause of this reduction in force is a decrease in the amount of calcium being released. So this amount corresponds to the normal amount of calcium that's released, so that's 1 micromolar. And you can see in the fatigue state here, then we're down to 0.5 micromolar. Okay, so that is going to contribute to this decrease in force production. Okay, so going from this amount of force to this amount of force when that amount of calcium is being released. So the amount of calcium being released is reduced. But there's another thing going on, and that's in a decrease in the sensitivity of the myofilaments to the calcium, meaning for this amount of calcium, force isn't reduced to this point from here, it's reduced even further. Okay, so the myofilaments are less sensitive to the same amount of calcium. Okay, so force is reduced because of problems with the going from here, this amount, to this amount. But there's also a change in the sensitivity of the myofilaments to that calcium. So force, if there was no problem with sensitivity, force would be reduced to this point, okay? But it's not. It's reduced to this point. And that's indicative of this sensitivity issue. And phosphate is going to cause that. Phosphate causes that. Hydrogen ions will cause that. Reactive oxygen species will cause that. Okay? So those are the primary things affecting the sensitivity of the myofilaments to the calcium. Would be phosphate, hydrogen ions, and reactive oxygen species or free radicals. Okay, this is another summary slide, probably even a broader summary slide, illustrating the fact that if you're going to fatigue, everybody agree, if you're fatiguing, it's because your intensity of activity is relatively high, right? And so if it's high, then that means you're going to be relying a lot on glycolysis, right? And we're going to change this to lactate, and so we will produce a lot of lactate, and that can be measured in the blood. There's going to be an increase in hydrogen ion concentration. There's going to be a decrease in concentrations of ATP in creatine phosphate, with decreases in creatine phosphate being much greater than the decrease in ATP. You're going to see increases in certain metabolites, such as ADP, inorganic phosphate, creatine will all go up. And as a result of that metabolism, then circa is going to decline, and that's going to be a primary cause of the reduction in relaxation, so that's important. Okay, so relaxation is reduced primarily because of a decrease in the activity of the pump that moves the calcium from the cytosol to the SR. A decrease in ryanodine receptor activity, that's the decline in SR calcium release. And so all those things, all these last few things are going to be manifested by a decline in force production, maximal force production. Okay, and so what you typically see then, again, increase in inorganic phosphate, increase in ADP, increase in free radicals or these reactive oxygen species, increase in hydrogen ions. So those are your key metabolites that increase, that impair the system. And we can add potassium as far as the T-tubules are concerned. And then you're going to have potentially substrate depletion. So this is what's going to be required to make the energy. And glycogen is going to be depleted, and ATP is dropped a little bit, but creatine phosphate is dropped a lot. And so other than glycogen, then it's generally thought that a decline in ATP or creatine phosphate isn't the primary cause of fatigue lasting on the order of 5 minutes to 10 minutes to 15 minutes, 30 minutes. Okay, it's usually going to be related to these metabolites that are increasing, and in particular, the inorganic phosphate. Okay, so increased inorganic phosphate associated with problems with the cross bridge, SR calcium release, and it can slow down circuit activity. Reactive oxygen species affect cross bridge activity, calcium sensitivity, and will contribute to slowed relaxation. And then what historically is thought to cause all the fatigue is now only considered to affect primarily relaxation, slowing relaxation, and impairing sensitivity. But those things will definitely contribute to a decline in power, as well as all these other things too. Okay, so inorganic phosphate and glycogen primarily affect the SR calcium release, and that's what's the primary cause of the weakness associated with fatigue. The hydrogen ions primarily affecting relaxation and sensitivity of the myofilaments. We're not saying that it doesn't contribute to fatigue, but it's now thought not to be the primary cause of fatigue. Okay, so can everybody define fatigue? Sure. What happens to SR calcium release during high intensity exercise, and how does this affect muscle power? So what happens to SR calcium release when the muscle is fatiguing? It goes down. So what happens to power? It goes down because force production is going down. Okay. Why is muscle glycogen affected in most competitive endurance races? Why is muscle glycogen affected in most competitive endurance races? The key word here is competitive. Well, you would, but would that be happening if the intensity was low? No. I mean, it would. Well, if the intensity is low, then you're likely not to recruit 2X fibers, right, which relies heavily on glycolysis. Glycolysis will take place in type 1 fibers, but most of the energy will be derived from oxidative phosphorylation. And so the key here is if you're competing, then your relative intensity is going to be high, right? So if you're trying to do something better than what you've done before, your intensity is probably going to be 70, 80, 90%. And so that's going to force you to use type 2 fibers that rely on glycolysis and muscle glycogen. And so those are likely going to be used, and the glycogen levels will be reduced. And when they're reduced to some critical threshold, then calcium levels within the cell are going to be reduced with each activation of the muscle. Everybody see that? Competition, usually if you're competing, you're going to exercise at a high intensity. If you're exercising at a high intensity, you're using all three types of motor units. If you're using fast motor units, you're going to be using glycogen. If you're using glycogen, they're going to be depleted. And so you want to do everything you can to try to preserve or spare your glycogen, because once it drops to a critical level, you will not be able to maintain your power output. Your performance will start to suffer. Okay, so how does muscle glycogen affect muscle power? Decreases SR calcium release, which decreases force. Force times velocity affects power. Okay, let's take a 10-minute break, and then we will start muscle injury. Even more slides associated with muscle injury than fatigue.